name is Hardeep Kaur. I'm assistant professor in RIMT University. Today's my topic is evaluation of crude drug. Evaluation of crude drug is very important in the case of crude drugs. What are the crude drugs? Crude drugs are those substances which are directly obtained from the plants, animals, which are directly obtained from the natural origin. So the evaluation is also very important factor in those substances because so many impurities are mixed in natural sources during the cultivation, collection, etc. So many processing. So how you determine the crude drugs? The various factors, various important points which covered in this topic. These are evaluation, my organoleptic evaluation, microscopical evaluation, physical evaluation, chemical evaluation, biological evaluation, and standard in the pharmacopoeia. Firstly, evaluation. What is the evaluation? It is the determination of the identity, purity, and the quality of the drugs which are directly obtained from the natural sources. So in other words, we can say the identity of the drugs which are confirmed by comparing the sample with the authenticated sample, or you can comparing the sample with the literature that is available. Main factor that affect the crude drug, that is quality. What is the quality? Quality, it is mainly amount of active chemical constituent present in crude drug. Second, purity, it refers to the freedom of admixture, free from making the drug free from unwanted impurities. Next is organoleptic evaluation. Organoleptic evaluations deals with the visualized determination of the drugs, that is covered the points which are size, shape, taste, odor, color, fracture, etc. And microscopical study is determine the microscopic characters of the organized drugs. We determine the microscopic characters. These characters are such like that: type of cell, tissues present in the cell, cell content like calcium, starch, oxalate, mucilage, etc. Because the microscopical characters of one to another drugs are different, we can mistakenly select in the case of organoleptic evaluation because some drugs are similar looking, but in the case of microscopical evaluation we strictly determine one drug to another drug. We strictly evaluate the drug from one drug to another drug. And microchemical tests are also useful in the evaluating a certain organized drug. Microscopical evaluation also covered the quantitative microscopy. Quantitative microscopy also contain a stomatal number, stomatal index, venous blood number, Palisade ratio. Stomatal number, it is the total number of stomata present in per unit area. Stomatal index, like same like that, venous blood number is the number of venous blood per square area. Palisade ratio, each and every plant is containing a different characters that are different from one to another plant. So it is a next the physical characters. Physical evaluation. Physical, it is mainly viscosity, swelling factor, melting point, specific gravity. Each in the case of the Plantico ovata, in that plant we use a husk to obtain the different type of drugs. So swelling factor is very important in the case of that type of drug, and melting point, each plant have a specific melting point, specific gravity and viscosity also. Some other physical characters are refractive index, Ash value, optical rotation, volatile oil content, moisture content. Firstly, after that the foreign organic matters. Some foreign organic matters like that heavy metals also mixed to your crude drug that impute the quality and the purity of the drug. So, pharmacopoeia have prescribed a maximum limit for the other part of the plant and other organic matter in the crude drug. We can say the pharmacopoeia have already recommended the maximum limit that approaches if our plant is approaches that limit, so we can further develop the further drug from the that type of drugs. But if our drug is not, if our drug is reaching the above the maximum limit, that drug is not suitable for the further processing. These then extraction. The extraction is the process, is the method to extract out the active chemical constituents. How we can extract the extractive chemical constituent, how we can extract out the active chemical constituent which are present in crude drug. Two type of methods: water soluble extractives and alcohol soluble extractives. Water soluble extractive are those which are active constituent comes out when dissolved in the water constituent, and alcohol soluble extractive oil are active constituent comes out when they react with the alkaloids and the glycosides. Next is the ether soluble extractives. Ether soluble extractives, what are these? These are volatile oils and resins. Volatile oil content, the which drugs are containing a volatile oil, example cinnamon, clove, etc. These are isolated by using a steam distillation. Ash value is also physical character, it is also physical evaluation. Ash value is the amount of inorganic matter which is present in the crude drug, that is that impure the quality of the drug, that highly affect the quality of the drug. The ash value of crude drug is approximately according to the prescribed limit of the pharmacopoeia. So, types of the ash value: total ash value, acid also insoluble ash value. Total ash value means the drug after placing into the furnace, then how much its weight, and acid insoluble ashes when the your extract is react with the acid, then after that how much amount of the ash is obtained. Water insoluble ash, the amount of ash that is dissolved in the water, and sulfonated ash. Next is chemical evaluation. Chemical evaluation, it is the determination of the active chemical constituent by performing a different type of assays. These assays cover the DPPH method, total phenolic content method, total flavonoidic contents, and total alkaloidic contents. Assessed by the acid base titration. Different type of acid base titrations are performed to evaluate the active chemical constituents. Non-aqueous and non-aqueous titrations. Next is biological evaluation. Before performing the preclinical and the clinical studies, we perform a in vitro in vivo studies, that is in some cases some crude drugs which are not evaluate, in cases we are not clearly satisfied by the chemical and the physical method, so we perform the physical evaluations. We performing a different type of assays that is making a fit for human use, like these assays are living tissues, performed on the living tissues, living organisms, animals, microorganisms, etc. A biological evaluation also covered the bacteria and fungus which are used to determine the quality, antibacterial and the antifungal drugs. And the second is the biological assay of vitamin D, and some in the cases of some chronic diseases like the anxiety and depression, etc. The guinea pig and albino rats are used. What is importance of the pharmacopoeial standard? Importance of the pharmacopoeial standard is, it is the it provides the drug the uniformity in the quality, types of the standards, structural standards, analytical standards, physical constants. Pharmacopoeia. What is the pharmacopoeia? Pharmacopoeia is the official code that containing a list of established drug. Importance of some important pharmacopoeia which are mostly used: the Indian pharmacopoeia, British pharmacopoeia, United States pharmacopoeia. Pharmacopoeia standards. Pharmacopoeia standard, it provides the limits that authenticity of the crude drug is established by reference in the given pharmacopoeias. So, in this class we learnt about the organoleptic evaluation, microscopical evaluation, physical evaluation, chemical evaluation, biological evaluation standard in the pharmacopoeia. That tells that mostly our focus is on the purity and the quality of the drug to make the fit for the human use. Mainly the our point our point that we already discussed in this class, that tells the quality, how to improve the quality of the drugs which are directly obtained from the natural sources by using different methods. Thank you.